Hi guys, Dr. B here. So we are going to continue with chapter 3 and we are going to start with the parts of a cell. In this lecture, we're just going to talk about the differences between something called a prokaryotic cell and a eukaryotic cell, and we'll get into a little more depth about what is in an animal cell. We'll talk about plant cells in the next lecture. So like I said, your assignment is to follow along with this lecture, fill in the slides, fill in the organizer you have on Google Classroom, and you will turn that in for your attendance. So some common features of a cell. You have a cell membrane, which encloses the cell. The cytoplasm refers just to the cell interior. Something called a cytoskeleton. So cyto actually means cell, so cell plasm. Cell skeleton, so microscopic fibers that help provide structure. Kind of makes sense based on the name, actually. We have ribosomes in all cells, and these are structures in which proteins are made. We talked about proteins last unit, so hopefully you remember some of those that we talked about. DNA is also found in both types of cells, prokaryotic and eukaryotic, but they are both different, and we'll get more into detail later on. So prokaryotes versus eukaryotes. A prokaryote, if you kind of break down the word pro is actually similar as kind of like pre, so it means before. Karyo means kernel, so it refers to the nucleus. So prokaryotic cells are cells that are quote unquote before the nucleus, before the kernel. So the official definition, it's a single-celled organism that lacks a nucleus and other internal components, which we'll talk about it in a few minutes. Typical size is 1 to 15 micrometers, which is really, really small. When you get to chemistry and physics, you're going to be able to convert these units, but I won't scare you with that yet. So prokaryotes, they have a cytoplasm again, so everything within the cell membrane, a cell wall that provides support and structure. This is kind of a key to know that they do have a cell wall. They have a capsule in some that allows it to cling to things. So it allows it to be effective against humans, for example, because say we're talking about a bacteria or bacterium, I'm sorry, and it's able to cling to your nasal passage or maybe your stomach lining, etc. So a capsule allows it to do that. A flagellum is a thread-like structure that protrudes from a cell surface, and it usually helps these prokaryotic cells move around. Something else that has a flagellum that's not prokaryotic per se, but it's a spermatozoa, and we talked about that. Van Leeuwenhoek discovered that way back when. We talked about how he discovered it too. So those things, spermatozoa does have flagellum as well. So prokaryotes, they have a nucleoid, which is a single circular molecule of DNA, and its cytoskeleton helps provide structure, is made of actin. So the reason why I'm kind of highlighting or underlining these at least is because this is what we're going to use, or some of the things we're going to use to compare it to eukaryotic cells, which is animal cells or plant cells. So you can see in this crude diagram, you have the flagellum in the back, you have the circular DNA, so this is the nucleoid. So the DNA is called a nucleoid. You have ribosomes, which deals with proteins, a cell wall, plasma membrane, etc. So again, keep in mind, there's no nucleus, there's no organelles, which are the little parts, the little internal components of a cell. It doesn't have those per se. And its enzymes and ribosomes, which we'll talk about what an enzyme is later on, ribosomes are free to move around in the cytoplasm. So what's the difference between that and a eukaryote? Well, a eukaryote has a cytoskeleton that's made up of something else completely. has a nucleus, and it does have these little subcomponents called organelles. And U, like Eucharist, if you will, stands for true. So true kernel, if you will. That's probably a good game handle, true kernel. I don't know. All right, cytoskeleton. So it's the framework of an animal cell. It gives it shape and stability. There are three types of fibers: actin, microtubules, and intermediate fibers. Actin helps determine the shape. Microtubules are the transport of information from nucleus to other parts of the cell. And intermediate fibers provide a frame on which ribosomes and enzymes can be confined to particular regions of the cell. So these three fibers make up a cytoskeleton, while prokaryotes only had one. So that's a good comparison that you should probably keep in mind. Hint, hint, nudge, nudge. So a cell membrane it contains the cytoplasm of a cell, and it has selective permeability. If you just break this down, if something is selective, means it's able to pick certain things, and permeability means it can allow things in. It's permeable. So a cell membrane has this capacity, and it's made up of a phospholipid bilayer. So this is what it looks like. This should look a little familiar. We talked about it in the previous units. So remember that fats or lipids have a hydrophilic head and hydrophobic tails. So these like to interact with each other, while the hydrophilic actually likes water. So it's going to form this bilayer. It's because it forms a bilayer because of these components: the hydrophilic head and the hydrophobic tails of these lipids. And selective permeability in this case means it will allow certain things in and maybe not let certain things out, or vice versa. So cell membrane proteins, there are a few to keep in mind. Cell surface marker, it IDs the cell type. The receptor protein is able to recognize and bind substances outside the cell. If you want, you can kind of think of these, I almost want to say like a lock and key. So this is like, hey, come over here. This receptor protein is able to recognize and say, hey, insert your key here, open it up so you can come in. Enzymes are proteins that assist in chemical reactions, and another protein is a transport protein, which by the name means it allows substances to move across a cell membrane. So the nucleus, it is surrounded by a nuclear membrane envelope. It stores the DNA. The DNA means that this is where things are controlled. This basically kind of starts everything. That without the nucleus, there would not be an animal cell, if you will. One, I guess you can say subcomponent of the nucleus is a nucleolus, and this is where ribosomes are partially assembled. So if you look, this is the nucleus, and then you can see that it has the nucleolus, nuclear envelope, and that from here you're able to make ribosomes, and that's where this part is going to come in, the endoplasmic reticulum. Like I said, ribosomes and the endoplasmic reticulum. So cells make proteins on ribosomes, and this right here refers to these internal membranes that help move proteins and substances throughout the cell, or through the cell, I'm sorry. It's made up of lipid bilayer, kind of like the cell membrane. So just imagine this, that inside of you right now, okay, what was that? Imagine that inside of you right now, there is all your cells are all doing this. They're making proteins, making ribosomes, and the DNA in your nucleus, in your cell, is actually helping, is kind of facilitating all of this. Sorry.